In animals, sex is determined very early on in development, at which point the subsequent developmental trajectory is locked in. Once sex determination is triggered, downstream maintenance and development is a long process that must continue correctly for proper development. In order to understand this process, we often look to genetic factors responsible for controlling this trigger. Let's zoom in on this zygote, which has a few weeks to go before sex determination takes place. Each cell within the zygote contains a complete genome. This is the collection of chromosomes within the cells. Each set of chromosomes have what are called the sex chromosomes, which can be either XX for female or XY for male. Interestingly, male or female fate is not decided by the entirety of the sex chromosomes, but actually just one gene. This is called the primary sex-determining gene, which is located on the Y chromosome. At the time when sex development has to begin, the male gene turns on. This directs other male genes to turn on in successive order, creating a positive feedback loop that results in male development. However, if the male gene is never turned on, neither are the downstream genes that support male development. This results in female development. My lab focuses on two closely related species of fish, the brook stickleback and the three-spined stickleback. Both species have XY chromosomes and a male sex-determining gene. In three-spined stickleback, this is guaranteed to cause male development, as expected. In brook stickleback, however, the male gene is only seen in males 85% of the time. This means that in 15% of males, they don't have the male sex-determining gene at all, instead carrying two X chromosomes. This means that there is something else we don't know about that is causing male development in these fish. Ultimately, I seek to characterize the differences between these two species to better understand this gene and its evolutionary trajectory. I also seek to understand what other genes are affecting sex determination in the brook stickleback.
Hi, I'm Katie, a third-year PhD candidate at the University of Georgia. Ever since learning about Darwin's finches in school, I have been enamored with the study of evolution. 200 years after Darwin, comparisons between species continues to inform our understanding of the world and how it operates. My work focuses on a group of fish in which the sex-determining gene has evolved multiple times independently. Using fish as a model organism, we can inform the basic understanding of how sex determination genes evolve, but this can also have implications in human disorders of sex development and infertility.
To characterize the unique patterns seen in brook stickleback sex determination, I will look across the genome for regions associated with maleness. Then, I'll identify when this gene and the known male gene is expressed. Once we know how these genes are being expressed early in development, we can turn to three-spined stickleback and eventually to humans to understand how these two genes interact in these organisms. This informs our understanding of sex development in both fish and humans and potentially how to better treat disorders of sex development. Thank you so much to the ARCS Foundation for this opportunity, and thank you for watching.